Well, we're back again to talk about tar spot today. We're going to discuss the epidemiology, model development, and decision support system that has been developed. With me today, I'm Darcy Telenko at Purdue University. I have Damon Smith from the University of Madison, Wisconsin, and then Martin Chilvers from Michigan State University. So this morning, we're just going to talk about tar spot and some of the things we're learning as we've continued this research on this disease. So I'm going to start with a little introduction of tar spot. As you can see, we have the tar spot life cycle here. We have shown that it is overwintering in the Midwest. We've confirmed this from data back in 2019 and 2020. And once that inoculum is there, the stromata, those black tar spots, release spores into the canopy. If the field has had a history, that disease can start from the lower canopy and move up. We do know that temperature and moisture are important for tar spot pathogen, with temperature being really important now as some of the data has come together. But the short-lived high temperatures are not lethal. And then as the disease develops, we have seen that it moves up in the crop canopy, and then those spores are also leading to a long distance dispersal, just like Fusarium head on wheat. And then once the disease develops, this disease is then shed back to the soil and available for future years. So we have multiple cycles occurring in each season. And as we talk more about the epidemiology, it'll all depend when the disease really kicks in and how severe it's going to be that season.
So Darcy, I have a question. We've done quite a bit of work on the overwintering of inoculum on residue. What do you think about moldboard plowing for residue management against this pathogen?
Personally, I would not recommend it. I think we've seen that it's surviving multiple years, and there's a lot of inoculum out there that I don't think plowing is really going to aid in slowing down the disease. Once it's in the field, I think this pathogen's present.
Yeah, I would agree with that. I don't know, Marty, if you have anything further
Yeah,
to add. I think, if you're in conservation tillage, don't abandon conservation
Mhm.
tillage to manage this disease.
yeah. The thing I'd add as well is like even if you were to moldboard plow, spores are going to be moving in from outside anyway, so it doesn't make a difference. We've had plenty of examples here talking to growers where they haven't had corn in a field for many years. They plant corn and it's been smoked with tar spot, so it just demonstrates how inoculum can move in from outside.
Right.
And,
Right.
and adding to that, Damon, I guess in addition to not plowing, I would also say rotation's probably not going to help influence this disease as well because of that movement of the spores.
Yeah, I would agree with that as well.
Yeah.
And so some things, if you haven't seen tar spot, what we're looking for as we're out scouting these fields is these small black raised spots. They can be circular to oval on those infected plant tissue, particularly the leaves, maybe even the husks of the ears. When you're looking for those spots, what I'm telling my growers is it really growing and embedded in that tissue? You can scratch at it, try to wash it off, and those initial lesions that develop. And then if you also flip the leaf over, you can probably see that stromata. That's what's called the name of that spot on that leaf on both sides of the leaf. And we can see it both on the healthy green tissue, and then as we have the tissue dries down and turns brown and blighted, they also can be found. And then some instances we might see that halo or what we call a fisheye lesion around that stomata. I don't see it as regularly in Indiana, but it is present at most fields at a low level.
Darcy, I think we should just talk really quickly just about other saprophytic molds and things, because people do get confused right at the end of
Right.
the
Yep.
season. Do you want to just describe that?
Well, I guess the one disease that might mimic it is southern rust. If it's late in season, we see something where if you look at that surface, this pustule is bursted open on that. And early on, we have a lot of false reports that are insect frass that could be, and that's why I say scratch at it and see if you can remove it, because insect frass will be removed. And then if you really haven't seen that full stromata develop, we may not be able to, could be something else in that leaf. So it's really important to make sure you have the right structures there to confirm it. That's what our diagnostic labs are here for, to help us validate whether the disease is present as well.
So Darcy, are we really concerned about this fungus getting into the seed? Is that a major concern at this point based on what we know currently? What are your thoughts on that?
Based on what we know, we don't believe it infects the seed. But where it's may be moving around is the debris, because it is surviving on any of that leaf debris. So that might be one way it could be moving field to field on those plants that are diseased. But generally, we don't worry about. We don't think it's infecting the seed. So that's as far as we know
Good
on that.
sanitation, good seed cleaning practices in the seed production end of things.
Yep, that'll help minimize movement through that way.
Very good.
And what we have here is just showing the annual progress of tar spots. So I think you've probably seen these slides before on how it continues to spread. So, it first started and we're first found in Indiana and Illinois and back in 2015. But as you can see, the disease continues to spread and it continues to move into new corn production areas. So it is important to understand if you still haven't seen tar spot, to keep an eye out for it, be ready for it. If it's not on your farm yet, maybe you don't need to worry about it yet or changing any management practices, but just be aware that the disease is still continuing to move across the United States.
Would you use this, for kind of watching out what's going on in season? I mean, I tell farmers here in Wisconsin, Darcy, that, this map's useful, I guess, to understand where it's been and where it's going. But as far as actually trying to make some in-season recommendations for us in Wisconsin, I mean, this map is not very useful for that because we have so much resident inoculum now that it's here, it's established, So I don't know what your perspectives are, in Michigan, Marty, or
Yeah.
Darcy, but that's kind of what I've been telling farmers in Wisconsin.
Yeah, I agree. I think for Michigan, yeah, you should just be scouting anyway. I've told them this season too, like I'm happy to put it back up on the map as we find it county by county this coming year. But I think it's more critical for those states that haven't dealt with it before, just being, as an early warning tool for those that just haven't seen it. I know,
Right.
Darcy, you've done some really extensive mapping as well in terms of severity across your state, which has been really interesting.
Right. And that's kind of why you see sometimes that Indiana fills in really fast. We're not going to continue that because it is a lot of effort. But if you went to go look at the maps now, anything that's yellow, that's appearing yellow now appears gray. So it tells you that where the disease has been previously confirmed. So now we know it's in that county. And what I tell my growers now is we're still going to be out scouting for those first vines to make a decision. And the decision is that it's active. So if I turn a county yellow in northern Indiana, that's probably where I'm going to be looking. It doesn't mean get out and spray. It means now we found active disease. It's time to really get out and look for this disease on your farm to then make an informed decision if we need to do something down the line.
Yeah, I like it. I like that a lot.
All right. So yeah, sorry, jump in. So what do we like I guess when tar spot broke out, we were all sort of jumping into the literature to try and figure out what was known about the disease already. And it has been sort of established in that Latin America area for about 100 years or over 100 years. Since 1904 was the first description back in Mexico. So I guess this Hock paper really describes what they found. So monthly average temperatures of 63 to 72 Fahrenheit. So we sort of figured it'd be sort of a moderate temperature disease. And then a lot of leaf wetness indicators. So average relative humidity greater than 75%, leaf wetness 7 hours or more during the night, and then 10 to 20 foggy days per month, and greater or at least about 5.96 inches of rainfall a month. So those were the factors sort of pulled out of the data that was available in Mexico and elsewhere in Latin America as to what was driving tar spot. We've definitely gone way beyond that, and Damon's going to jump into what we know now. Another really interesting thing is sort of taking some of these parameters that were thought to sort of be predictive of tar spot. The group in CIMMYT in Mexico pulled up this risk prediction map. So basically some weather comparison between what we knew of the weather parameters predictive of disease and what they expected in the US. I think we can say now that the map's good, but it's tar spot spread beyond this. It's in Florida, Georgia. And I'm kind of curious how it's going to perform in irrigated areas too, like Kansas, given our experience. Darcy and I have a lot of irrigated acres here in Indiana and Michigan. And so it's been pretty eye-opening to see what can happen under irrigation. But that's what we sort of knew. So Damon, spotlight is yours.
Yeah, so we've kind of built on this a little bit over time, and there's been some groups that have been looking at this sort of in controlled experiments. Actually, one of your old grads, Marty, has been looking at this with Bayer Crop Science, and this was a really interesting study. So they were interested in trying to get a controlled inoculation to be successful, because it's been really hard for this pathogen. And the idea being, if they could get that going, they could start to screen lots of hybrid or inbred material to try to find some resistance. But from an epidemiology perspective, this paper was really interesting for me and my group, because what they found here is that their most successful inoculations were actually where they had some early continuous wetness and kind of moderate temperatures in the first 5 days after inoculation. And then they actually had to withhold misting and wetness. And this was, sort of counterintuitive to what we know about a lot of these foliar pathogens. But apparently this particular pathogen, at least in controlled experiments, and this particular isolate, at least against the hybrids that they were at least looking at, what this was saying is we needed wetness right up front after that infection to probably facilitate that infection. And then we actually had to withhold wetness afterwards, just have some high humidity there in terms of actually having disease progress. And this was kind of a breakthrough for our group, because we had seen some similar things in the field. We were actually looking at field data, what had been going on since the 2018 epidemic. And we were seeing that these sort of intermittent wet dry cycles were kind of important from a field perspective. And this kind of corroborated, although I know, Marty, in your program, you've kind of seen some slightly different reactions there. And I don't know if you have any comments there on that.
Yeah, basically that we've managed to get tar spot to develop quite nicely in the greenhouse under essentially continual misting, not continually all the time, but hourly. So we really need to pull this apart a bit more. And I think, Damon, part of the challenge too is monitoring, measuring leaf wetness.
Right.
Such, what does my misting mean compared to their misting system? And there's a lot of questions that we need to jump into. And maybe it was a different strain that resulted in some of the differences, but certainly I was so happy late last year, and I'm showing all the growers this extension season, like some of the images of what we got going on, because it was so challenging. And I thought my students were doing something wrong for the longest time. I'm like, oh, let me get in there and show you how to do it. And I couldn't do it either, right? And so we all struggled with that.
Yeah, we've all, my program, Darcy's program, we've all tried at it. So it's great that we're starting to get some breakthroughs here. One of the other pieces here was that this group said that the incubation period was 15 days. So the incubation period is the time from when that spore infects to when we see the first tar spot develop, and then the latent period, which is the time from infection to when we start to get the repeating cycle or actual spore formation out of that new stromata. And so this was interesting, because this was starting to shed some light on, how long does it actually take to get from that infection to when we actually see disease. And we'll talk a little more about that here as we move along. So I want to just kind of step back a minute, though, and kind of talk a little more about, how did this thing actually get here, and now consider what's going on here in the Midwest now that we've sort of laid the lay of the land in terms of what it takes from a moisture and temperature perspective. And I'm just going to drill in here to Wisconsin for a second and just pull this out. But this is really kind of going on, I think, across the upper Midwest in general. Probably really, we're looking at the same trend across the Corn Belt here in the US. But what we've noticed here in Wisconsin is that, our temperatures are getting warmer. Our temperature departures are 2 to 3 degrees Fahrenheit warmer than they have been, over the, 50 years previous to now. And with that, where we have warmer air, we can obviously hold more water. And so we've also seen a trend for higher rainfall per season in general. I mean, we can still have drought seasons and that sort of thing. So we're trending wetter. And what's even more interesting, I think, for my perspective as an epidemiologist and somebody who works really intensively in disease forecasting systems, is that we're seeing these larger sort of heavy rain events. So in Wisconsin, we consider a 2-inch or more per event in precipitation as a heavy rain event, and we're seeing more of these. And anecdotally, we've sort of noticed this over time, where it'll be really dry, and then we get really wet, and then we get really dry again. The seasons where we have kind of a nice inch of rainfall every week, we just don't have that anymore. And so the patterns have just changed. And I stepped back and kind of looked at 2018 here, and across the top we have average, temperature across these top panels starting in June, then going to July, August, and September. And of course, we're trending a little warmer, and we've learned that this pathogen doesn't really care if it gets too hot. But more importantly, what we saw here is that June, we were kind of wet generally across the state, but then we turned very dry in July, and then got extremely wet again in August. And this type of wet, dry, wet pattern seems to really line up. And we looked at it again in 21 in Wisconsin as well, and we saw the similar sort of trend. So this was starting to just, again, this is sort of anecdotal evidence, but starting to corroborate what the folks at Bayer Crop Science had seen in their control inoculations. And I know, Marty, I think you've seen maybe some similar things in Michigan, but maybe even a little bit wetter in Michigan.
Yeah. This is so interesting, right? Like what is really needed in terms of moisture and dryness and whatnot. Yeah, so I mean, I still feel like even though we've done a lot, we've still got a lot to sort of learn, right, about exactly what's driving things, exactly what we mean by leaf moisture.
Right.
I mean, but without a doubt, like 2018, 21, where we had, fairly frequent rainfalls, and there may have been dry periods in there too, in Michigan as well. Like we just had explosions because you had disease set up early on and then just continue to rip through. And I get the question this year, like, why didn't we have tar spot? So, by the end of June, we were really wet. We had heavy rainfall events throughout the rest of the season, but we missed that really initial build up, that initial infection from residue. And I think that's why we just didn't get that initial build, right?
Yeah, things just escaped. And I don't know, Darcy, I think some of this has to do with just hybrid reaction and the interaction of certain local, I don't know if it's an ecotype or isolate or what we want to call it, but the pathogen side in combination with the hybrids we're growing. I don't know if you have some thoughts on that.
Right. And I think we shared with some other talks about the hybrids. So we know that there are some significant differences between their hybrid reaction and how fast it develops in the canopy. And also, trying to get good ratings on hybrids is what we're having problems because some things we see in Indiana look different in Michigan or Illinois or other states. So again, it may be the differences in the populations we've had that were introduced. And then what's changing. And then I would say, so back on your temp moisture conditions, I mean, in Indiana this last year, if I look at it compared to the other years, we started off a little wet and then we went dry for a long period. So even though we did have some early reports, it went so dry that we stopped it. And then we were wet in August again and it kicked back in at the end of the season. But then the question is it late enough that we're not going to impact yields or not? And then if we look backwards, 2022 was so dry that we didn't have any disease hardly at all. And so again, it's that I think that weather is going to be really play a role in how fast this disease gets started in each season and whether it continues.
Yeah, in Wisconsin, we had a similar thing happen where it just got, it started, then it got so dry, it just couldn't move. And then we sort of escaped at the end of the season. So I'd like to just kind of step in now and look at the model development that we have. And this work was supported by partially by the National Predictive Modeling Tool Initiative, which is administered through the USDA. And what was great about this program is it helped us sort of coordinate large data sets. So we had multi-state trials. We actually had 246 site years, and these were a combination of both small plot and commercial field experiments. And you can kind of see the breakdown. The blue dots over here in the national map, these are overlaid on the estimated acres from the USDA in terms of corn planted. And you can see blue dots, commercial fields, red dots, small plot trials. So we actually had quite a few more commercial fields, which was nice in this particular data set. This all amounted to 588 individual disease observations over a period of time dating back to 2018. So we had heavy epidemic years and lighter epidemic years. And if you really want to geek out and talk science, this has actually been published as of last fall, and you can actually go out and find that at Scientific Reports at that link below if you want to read and just see what the science looked like. Just to give you a little bit of lay of the land in terms of how we sort of coordinated this. It's hard to get everybody on the same page without some visual diagrams. And one thing that came out of the Crop Protection Network a couple of years ago is we sat down and really coordinated what severity ratings look like. And so these are computer generated. Those are pixel counts. So these are true 5%, 10%. 50% coverage. We also can rate the eye spots, so you can see a separate set of rating scales in the lower hand. I'll say, at least from my perspective, we mostly see The tar spots nowadays, we don't see too much of the fisheye lesions anymore, and that could be related to hybrids or just, weather and that sort of thing. But, these were nice because we could hand these out to all the collaborators and we were all on the same scale, right? And so that was the first big hurdle, was just trying to get a common rating system. And I think the other big piece was having some uniform trials and uniform data sets that we could then go back to and start to pull all this data. So just to also show you kind of how intensive the rating was, we had trials set up. This is just one location, Arlington, Wisconsin, where we had basically two types of trials where we had corn on corn, no-till. So we had high residue here, trying to get that inoculum load up. We were rating through time, so starting at tassel, going all the way on through to almost full maturity here. And then we split the canopy in a lower, mid, and high, and we actually had two different hybrids, a resistant hybrid and a susceptible here. And you can kind of see the ratings, and these were intensive, 2 to 3 times a week on these small plot trials, a couple times a season. In the commercial fields, we didn't have all the layers in terms of treatments in the commercial fields, but we were out there rating it a couple of times each season. And you can see how the disease kind of spreads from bottom up in the canopy. So we learned a lot about just in-season spread. And we also learned, a lot about residue. And we talked about residue a little earlier, and this is a case where we had, same location. This was a different field, a little ways away from the previous field. This was soybeans tilled, so really no visible corn residue, and we still had tar spot increase. Yes, some slight reduction in terms of increase, but we could still see it move up the canopy and get pretty heavy on that susceptible hybrid here. So I don't know, Darcy, Marty, what are your thoughts on where the heck is the inoculum hiding out if it's not on residue in the field here?
Well, I think it's still on the residue. It's just microscopic. If you think about how small those stromata are, we can't really see that those leaves are still there, but those stromata are. And I think that's where it's hiding in, is it's still there. If you think about the millions, billions that we're shedding back to the soil every year, we only need a few really to release some spores to get it started.
Yep.
Yeah, it's amazing how much of that leaf debris will break down. But if you really start digging in the cracks and whatnot, you can find some of those stromata structures. It just takes a lot of digging.
Yeah, and then, again, this is why I don't think you should abandon conservation tillage just to manage this disease. It's just the inoculums here. It's an established disease here in the upper Midwest for sure. Mm-hmm. So, once we had all that tar spot data, we then, had to actually blend the data together with some weather data. And we were using IBM historical weather. So this is a grid-based weather system from the cloud. So we had GPS coordinates for all these commercial fields and small plot locations. We take that weather data, we combine it with the disease data and put that together. And then we can do things like correlation analysis and actually look at window panes. And the window panes are useful from a biology perspective when we're trying to do this modeling, because it helps build in, what is the incubation period? What is the latent period? So we actually were looking at 14, 21, and 30-day window panes. So we're actually pulling weather over time for a number of variables. We generated lots and lots of variables, enough that we could actually generate 54 logistic regression models. And we also experimented with some machine learning algorithms, including random forests and artificial neural networks. Once we settled on some of the better models, we actually were able to integrate those models into some testing platforms, decision support system testing platforms, and we call that tar spotter. So we actually went through, I believe we're actually at 5 iterations now of tar spotter. So this is something that's an iterative process, goes over time, but I think we've settled on something that's really quite good. So again, published data, if you really want to kind of dig in and see what we're doing here, you can get an idea. These are the correlation analyses. So things that are in blue are positively correlated with disease. Things in red are negatively correlated. And you can see the number of individual variables that we're actually looking at. So we have 30-day variables up here in this pane, 14 here, and 21 over here. So large number of variables that we were looking at to combine with that disease data. We were able to run that through our logistic regression modeling. work, which basically is probabilistic modeling, and then also the machine learning algorithms. We actually generated 8 really high performing sort of more traditional logistic regression models, which we call LR1 through LR8. And then we actually combined a couple of data of models together into what we call a multimodal ensemble data set here. And then you can see the random forest and artificial neural network models here. But what we did is actually adopted the multi-modal ensemble. And the reason why we did that is that really balanced accuracy with errors. So that's really what we want to do. So, we don't want to err too much on the spray side. We don't want to err too much on the, hey, we missed a spray side, but we're also trying to be reasonably accurate in terms of the number of times we're successful. We're able to actually measure that accuracy by partitioning those 588 observations we have. We actually take 70% of those as a training data set, and then we save 30% actually as a validation data set. So when we go through this whole process, we randomly pick those, and so you can see we're pretty good. We're pretty close to the random forest models here in terms of accuracy, but we better balance really type 1 and type 2 errors here. So that's why we chose those models. And those models also are a little more straightforward in terms of programming into a disease support system. So if we were going to actually look at what the 2 individual models might look like on a response surface here, this is the response surface for both models. So we have risk probability from 0 to 100% risk on both of these vertical axes. And you can see we're heavily weighted in terms of moving average of mean daily temps, 30-day, and then 30-day maximum relative humidity is actually our wetting parameter in this particular model. And then we have 14-day moving average nighttime hours greater than 90%, which is sort of a leaf wetness surrogate approach here, and then a 30-day mean temp occurring again. So we get the risk probabilities from both of these models. We put those together, and then we take an average response from those and actually report that probability out. And again, that's all running currently in the current version of TAR Spotter. So the interesting thing, once we had published this data, and we actually made an argument in there about actually in the field that incubation period might be just a little bit wider, maybe more like 21 days, and the latent period might actually be closer to say 35 or 40 days. And there's been some work. Dean Malvik and his students at University of Minnesota just actually published this paper looking at temperatures and incubation period and wetting events kind of in some controlled field inoculations, interestingly, and really corroborated kind of what we all been seeing. We have to have sort of these mild temperatures to really get things going. And they actually report sort of these intermittent wetting events as well there, but their incubation period reports were kind of on par with what we saw through our modeling work. So 20 to 22 days is what they were reporting there. And then, closer to 40 days in terms of the latent period. So the time from when we actually have inoculation and when we start to see that secondary cycle really start to pick up. So I think, I don't know, Marty, Darcy, I think we're all starting to see some patterns, at least out of all this different work where we're looking at the biology. We're also tying that to what we're seeing in terms of weather events and all that. And I think we're starting to get a little bit of lay of the land, at least in the upper Midwest here, in terms of what it actually takes to get this thing going. I don't know if you have any comments.
No additional comments. I think you've done a good job of
laying
that out. I think, like you said, we're all seeing this similar sort of thing with some of the work we did as well with some of the genome work. It took that 21 days really to see symptoms start to develop. So it's just this common theme, which is nice, right? It's helpful to have some confirmation.
Yeah, and I think it also explains, in terms of management, why, if you wait to see it, you're so far behind the 8 ball, so, really by the time you're actually seeing those stromatolites, those infection events probably happened 15, 20 days earlier, right? And so, this is something that we really, have to sort of predict, look into that crystal ball and really kind of get it right. So we're actually have used TAR Spotter now. We've tested it a few times out in the field just to look at how do we actually apply you know, the tool to actually, you know, recommend fungicide applications. And you can see here, we did some scouting here in Wisconsin. This was actually a two pass. This was 2021. So this was our last bad epidemic in Wisconsin. You can see how well just scheduling the same fungicides, you know, in that right-hand image, how well it worked against the non-treated control there. So just to show you where you can actually find the tar spotter tool. There's actually two places. So the first iteration was tar spotter. So that was our first decision support tool. And then the UW actually helped us spin out a startup company called Field Profit. So Field Profit actually adds some bells and whistles there, and Field Profit version is actually now free to download as well. So you can find that platform out there on the App Store. The nice thing about Field Profit is it actually gives you a 7-day true forecast here, and we actually do these trend lines, so you can actually look at also what's happened over the past 7 days, kind of in real time once you set up your field. And we find that, you know, looking at the trends over time is quite useful in terms of, you know, being able to sort of anticipate, you know, is it really bad today, or is it actually trending downwards, right? And so if you're a little above that action threshold, let's say 40%, but the overall line is actually trending downwards, then you might hold up and just kind of pump the brakes a little bit, especially if it's the first application. You really want to maximize the efficacy out of that first location. So just to kind of take a look how you might set the tool up here. This is the Field Profit version. Just setting up a field here, and the tool is actually set up to hit the GPS on your phone, so it actually sees that GPS coordinate. Will actually choose the corn tar spot model. We actually run multiple models in the Field Profit version, or you can actually go to a different location. So you could actually be in the office. You don't actually have to stand in the field to get this. And we'll actually go down to Florida and look at maybe some sweet corn production here in Florida in the wintertime. Drop a pin, we set the field. So we're actually going to add that to our history, and then we can run a forecast. And we have our action threshold set. We answer a couple of questions about fungicide and growth stages. Then we run the model, and boom, we have our risk right away. So the phone actually went up to the cloud. It pulled down 30 days worth of weather data for that GPS coordinate and did all the math that quick for you. We're actually going to do a 7-day forecast now. So here's what the forecast actually would have been for that particular date. And you can see quite high. So you can actually look ahead and get a true forecast out of this particular tool. And then it's also saved in your history, and you can actually back out and kind of look at, you know, what's going on across the landscape in real time. And you can even set the phone to do a snap to refresh. So on a daily basis, you could go into your field list and just snap that list down, and it'll actually auto update your entire field list for you. So kind of a handy tool there to look at. And I'm going to kind of turn this over to Darcy to kind of talk about some work that she's been doing on timing, and then also incorporating tar spotter into some actual fungicide application trials that she's been running.
Right. So Damon, what you're showing here, what we had done is we had a timing trial, and I think we ran this across the region as well, but in Indiana, we actually had good disease develop. And so what we had is both a susceptible hybrid and a resistant hybrid, because we had those questions of, is the timing going to be different between the two hybrids if we have some resistance built in there? And I think this will show that maybe resistance is still important to make sure we have that as a backup first, and then look at our timing. This data did use the most recent tar spotter app that we had running last season. And so we went with the standard application timings. We started with a late vegetative at V10, VTR1, R2, R4 were our timings based on the growth stages, and then the tar spotter app. And for this year in Indiana, it triggered for that app to spray at R2, and then there was a follow-up, so that you actually had 2 applications, R2 followed by about an R4 application is where the tar spotter treatments were applied in season. And as you can see, you know, some things here, you know, our susceptible hybrid had significantly more disease than the resistant, and then we did see there was an interaction going, so that the timings weren't acting the same in each of the hybrids. And if I look at the susceptible in the blue, you know, most of our applications did reduce tar spot, with the tar spotter, that double application, providing the best disease control. These, I will say, we're all using a Delaro Complete in this trial. So I think in some of our work, we've shown it's going to vary depending on your product, the timings in each season as well. But this one's using Delaro Complete. And then if we look at the resistant hybrid, it almost shows that, that those earlier applications weren't really necessary in that resistant hybrid. They didn't provide the disease control as compared to the non-treated. Later applications, and I think there maybe it's a plant host interaction, something switched in the crop canopy. And I think I see sometimes our resistant lines start seeing more disease ramp up as we move into those reproductive stages as well. But you can see then for the resistant hybrid, we really didn't need a spray to that R2, R4. And again, the tar spotter still worked on that double application in the resistant hybrid. Damon, we don't have the yield data here, but I would say if I look at the yield data, because that resistance is holding some of that disease back, in our non-treated, in the susceptible hybrid, that tar spotter app was the one that did significantly bump up yield potential there on providing yield protection as compared to the non-treated. So for this year, I only had a few areas where we had a good fungicide response and yield response to a fungicide application with tar spot since it moved in so late. Whereas the others, fungicides were there, they control disease, but they may not get that yield potential. And that's the big question, is the return on investment for protecting against that disease.
Right. Yeah, it's a lot of work still being done, but I think we made some headway, at least trying to understand what drives us, at least in the Midwest. And I do think it's a little different, here than maybe what folks are seeing in Mexico. There's
Right.
some similarities, some differences, but we definitely need to keep shoving forward. So there's lots of folks we need to, of course, acknowledge here. We had numerous cooperators on the National Predictive Modeling Tool Initiative, who you can see all listed here. A lot of university support at each of those institutions. And then, of course, our programmers, Roger Schmidt and Terrell Schmidt, helping us with tar spotter development. In addition to the National Predictive Modeling Tool Initiative support, we had a lot of support from various corn commodity groups, state boards, as well as the National Corn Growers Association. That included Indiana corn, Michigan corn, as well as Wisconsin corn, of course. And then the Foundation for Food and Agricultural Research also helped.
Okay. And Damon, in addition, so you mentioned the really nice research article that's out there and is free to download and look at. That's pretty dense, good nighttime reading, something to put you to sleep. But we also have sort of a boiled down extension versions that are available on the Crop Protection Network. So I encourage you to have a look at that website for those. Included there are the fungicide efficacy work that Darcy's done a lot of leadership on. Some really nice articles just summarizing what we've seen, and those fungicide efficacy charts too, actually, that relates to that work.
Yeah, I would say I would add to that, Marty, that there is an assortment, even your work with the population and nitrogen as well. As we develop some of these research publications, we're translating them into an extension pub that's easier to understand. And I'd say we're looking forward to one on the efficacy. There's even going to be a bigger one that we're developing right now that should be coming out too in the near future. And I just also want to acknowledge that some of this work, and including these videos, is also supported by the USDA CARE initiative as well for this project.
Yeah, and in addition to all the extension work that's continuing on, we also have quite a bit of research. And I know, Marty, Darcy, you guys are looking at irrigation, and I'm anxiously awaiting, results of that, because we'd love to build that into, a tar spotter and those sorts of things, to really try to understand what's going on in those types of environments. Marty, you guys just, Darcy mentioned that nice work you guys did on nitrogen and also plant populations. So folks should go check that stuff out. But, all this work is ongoing, and we hope to continue to contribute to the knowledge base here.